It is now my honor and privilege to turn it over to our guest speaker, Dr. Shirley Levine.
Thank you, Kayla. In the beginning of June, I received an email from Kayla letting me know that there were going to be a series of seminars over the course of the summer and the fall, and asking each of us whether or not we had anything that we might contribute to information about COVID-19. And as a hematologist and internist, I thought for a while, I'm not an infectious disease specialist. I really have no special education in epidemiology. But as the days went by, I remembered how many clinical trials I had participated in. In hematology, I was in charge of, and sometimes a participant in, a number of clinical trials for cancer therapy. We took care of leukemia, lymphoma, myeloma, and the clinical trials were a very important part of practice. And it dawned on me then that any review of the clinical trial system would be tremendously important to any of us who are living through the development of vaccines or drugs for prevention and treatment of COVID-19. So I discussed it with Kayla and with Denise Wagner, and we decided that it was an appropriate topic. And I have had an enormous amount of fun, hard work, but fun, keeping up with probably what is 50 to 100 different press releases and newspaper articles about what's happening in both vaccine development as well as drugs that are being tried. So I hope to spend the first half of this time together really talking about clinical trial process in depth, because I think that anyone who's interested in medicine or any of the allied fields of medicine will have an opportunity to participate in clinical trials as an overseer, maybe as a developer of some of the clinical trials. But I also think that anybody who's just living needs to understand how to assess the many different reports that you are seeing. Within the last 2 weeks, I have seen 2 articles that were published as preliminary reports, and then they were picked up by multiple news sites. And one of them was about hydroxychloroquine, which, as you know, has been a major topic of discussion this week alone. And the other one was about patients that had significant respiratory problems that were treated with steroids. Neither of them fulfilled any of the criteria for a good clinical trial. And yet they had been published, not in first-rate journals, but they had been picked up all across the news. So if you're at all paying attention to the news that is being published, either for vaccines or for drugs, I think that what we will cover in the next 20 to 30 minutes will be something that will allow you to really look at those trials very carefully. After going through the clinical trial discussion, what I would like to do is to walk through 2 different types of clinical trials, or the things that you need to think about. The first one will be a vaccine, and I've chosen one vaccine, and I will tell you why as we get there. And we'll go through exactly what has happened in the process of developing that vaccine, from the moment that it started until literally a week ago when the very first preliminary report came out. The rest has all been business discussions. I know how much the stock goes up, the stock goes down every time they put out a preliminary report. And I think that that's an important thing for us to know about also, is that a lot of the information that comes out has to do with exactly what is happening in the financial world in order to maintain the ability to develop these vaccines. And then the second are 3 different, 2 drugs and one which I call really hopeful developments, but that's not much known about. But I wanted to end on a high note with some things that we may be able to look forward to. When I was in training, as I mentioned, we had a number of different clinical trials that were going on. And the basic fact that you need to know about any, either vaccines or drugs, is that every single vaccine and every single drug has to go through exactly the same process. Kayla, can you start my slides or am I? Yes?
Yes, I'm uploading them right now.
Okay. Maybe go back to the first one.
Okay.
Good. Okay, next slide. And the next slide. So every vaccine and drug must go through the same procedure to get Federal Drug Administration or FDA approval. It makes no difference what the vaccine is or what the drug is. They must fulfill all of the criteria that are set by the government. And not only do they report what trial they are starting, and any of those can be found on our website, which are clinicaltrials.gov, but they have to submit proposals for each phase, the 3 phases of the drug approval or vaccine approval process. Next slide. There are a number of different trackers that are available on different websites. The New York Times actually has a very good tracker. This is not from that, but I just wanted you to have some idea of the magnitude. And each of the numbers on this little pie chart that I made are a week old. So the beginning of this week, on Monday, they were all about 10 to 15 numbers higher. So in the vaccine category, there are 175 different vaccines that are being developed, and they will slowly enter the pipeline for approval. There are about 200 different antivirals at this point. And the bottom section where you can't see the number is 275 other treatments. So drugs that are not antivirals or vaccines fall into that broad category. So you can just imagine how much information is going to be coming out. And as a consumer, I think all of us really need to be aware of exactly where in the process each drug or vaccine is at the time that you're reading about it. Next slide. So my specific objectives, as I started to mention before, is to review the preclinical and the 3 phases of clinical trials for drug administration approval, FDA approval. Secondly, to trace the pathway for vaccine development. And I'm going to use as my example the vaccine that is being developed by Moderna. It was a vaccine that was developed with NIH, and NIAID stands for the National Institute of Allergies and Infectious Diseases. And that is the institute that Anthony Fauci heads for the NIH. Third is going to be to trace the pathway for drug approval. And the examples that I've chosen are dexamethasone, remdesivir, and to now, those are the only 2 drugs that have demonstrated any efficacy at all in COVID-19. And then last, for a little bit of hopefulness, I'm going to talk about monoclonal antibodies and hopefully be able to convince you that we should be hopeful, that things are happening. Next slide. So what is the standard procedure? And I'm going to first just go through it very briefly and then go into each of these phases. There are preclinical studies. If you have a vaccine, it is expected that you will do a certain amount of animal testing, sometimes mouse models. You'll see in the literature that some of these vaccines have been tested in non-human primates. And with the speed up of vaccine development for COVID-19, some of these things are being done almost simultaneously with the development of the protocol for phase 1 studies. Once they have some evidence that this is a beneficial, perhaps successful looking vaccine, they will go into phase 1 studies. And the purpose of a phase 1 study is just to assess the safety of the vaccine. These are usually small studies, anywhere from 25 to 100 volunteers. And the volunteers for vaccine studies need to be healthy. The drug studies, the patients really have to have a known diagnosis, a confirmed diagnosis, because you're looking at the effect of a drug on a specific disease. Once they have completed phase 1 studies, they go on to phase 2 studies, again with notification of the FDA, what that trial is going to be, and some approval by them to start phase 2 trials. And that is also to assess safety. But this time it's in hundreds of volunteers. And efficacy is and may be part of the data being collected. Now, in the phase 1 trials for the vaccines, there were some small number of volunteers that were pulled out, and they actually did look at antibody development in those individuals because they really needed to demonstrate that there was stimulation of the immune system and antibody production. And in phase 2, efficacy becomes an important part as well. But they may not do it in the hundreds of different volunteers that they have in phase 2 studies. And phase 3 is when the big trials take place. Thousands, tens of thousands of volunteers. And they're still assessing safety because they want to know about some rare side effects that may only be seen when you reach 20 to 30,000 volunteers who are being injected. But mainly it's efficacy. Next slide. So when they start off with phase 1 trials, the usual vaccine takes about 7 to 10 years to develop. Some take much longer. So some vaccines have never been possible. The primary one is HIV. There have been people working on vaccines for HIV for a long time, and so far, nothing. The record is 4 years, and that was for mumps. So what they are now setting off to do in the COVID-19 vaccine trials is something that has not been done before. And this is going to be possible based on some of the different developments of technology. Also possible because they're overlapping things a little bit. They may not have finished their phase 1 when they got approval for the phase 2, et cetera. So we may be lucky. And I think that a realistic goal would be anywhere from 12 to 18 months, as opposed to the record-breaking 4 years for the development of the mumps vaccine. In phase 1, they can test multiple groups, and usually it's by age, so that they are testing volunteers who are 18 to maybe 40 years old, and then 40 to 55 years old, and there may even be an older group because there's always a concern that the older population doesn't have an immune system that is as robust, and they may not have the same type of antibody response. So that can be groups that are based by age. Next are different groups based on dose, because one of the things that they're trying to do is to determine the maximal tolerable dose. So they can choose a dose to use for the subsequent studies. In the vaccine trials, as I mentioned, it's healthy volunteers. For the drug trials, these need to be volunteers with target disease. So if they were looking at drugs for the treatment of COVID-19, each of those individuals would have COVID-19, documented COVID-19. If it were drugs for a disease such as lupus, they would have to fulfill the criteria for the diagnosis of lupus, or for a bacterial infection, be able to have said that they've had cultures that have cultured out those organisms, et cetera. So in drug trials and vaccine trials, you're really looking for different volunteers for the trials. The subjects are monitored for changes, and they have symptom profiles that they need to fill out on a regular basis. They have physical exams on a set schedule so that every so many days they will be examined by some of the trial participants and overseers, and they will have various and sundry lab tests drawn, pretty much the kind of things that you would get done on an annual physical exam and lab test. They want to know what your kidney function is like, what your liver function is like, all about your blood parameters. So all of that is set out in protocol before the study even starts. And they may have different additional endpoints. In vaccines, obviously, the best example is antibody production. And they're looking for actual soluble antibodies, and they're also looking at how T cells are stimulated in the immune response. So all of that is happening in phase 1. And then when they have enrolled all of those individuals and injected most of them and followed them for some period of time, a protocol is then approved based on their phase 1, not necessarily all their phase 1 data, but their phase 1 response, and they are approved to go on to phase 2. So phase 2, next slide. The main goals are not only to follow safety, but really to select an optimal dose range. And for this study, they may still be using two different doses, until they find the dose that has the right balance between the efficacy that they want as well as the side effects that they don't want. And those are the main goals. Usually, certainly almost all the time, subjects are randomized. And that's for vaccine trials as well as drug trials. And the treatment group and the placebo group really should match as much as possible. So every characteristic of those volunteers is recorded and compared. They don't want anyone in the placebo group who has more severe kidney disease, or someone in the vaccine group who has heart disease, and that the percentages don't match from one group to another. And that's the purpose of randomization. There are tables of random numbers. I'm sure there are lots of computer programs for random numbers, and all of the assignments are controlled very centrally in all of these protocols. One person or one or two people control randomization. You want to eliminate bias. They're usually what's called open label, meaning that everybody knows what they're getting. The group that may be getting placebo may drop out of the study. But usually with these studies, at the end of the study, those that had gotten placebo will be offered the drug or vaccine. They are very closely monitored for an extended period of time to study the efficacy and the adverse effects. And they may break out a cohort to really look at efficacy in subgroups, maybe all the subjects. So in a vaccine trial, they may say that of the hundreds of volunteers that they have, they may take 50 or 100 of those and do efficacy studies, meaning antibody production, et cetera. Next slide. Phase 3 is really the big test of how good a vaccine is or how good a drug is. And the subject group should reflect the target groups. For a vaccine, that would mean that they want to be able to vaccinate everybody from age 18 to 80 plus, 90 plus. Children are usually done later, and they're done as a separate group all to themselves. But you can't offer vaccines to someone who is age 90 when they've never been tested in that group of patients. Subjects are randomized, again, between vaccine or drug and placebo. And it's really preferable that these are blinded trials, usually double blind, meaning that neither the volunteer or patient or the people running the trial have any idea what they've got. Now, sometimes it's a little bit hard to hide because patients that are getting drugs that may have different shapes, different colors, et cetera, they try to make placebo pills look exactly like the effective pills. For injections, same color of solution in the injection, if they're visible, et cetera. So blinded really means that you cannot easily determine which you have gotten, the placebo or the active drug or vaccine. And again, they are monitored for a prescribed length of time on a schedule to determine effect, duration of effect, and adverse effects. And having participated in many of these trials, primarily for treatment of cancer patients, these protocols can be 50 to 100 pages in length. And every single thing that could happen to the patient has a response that you may want to take. For example, if blood counts drop, you have to know how far they've dropped, and then there's some modification of exactly what the next step is, especially for drugs itself. You may actually, for a cancer chemotherapy drug, or even a drug for standard heart disease or lung disease or kidney disease, you may actually have to stop the drug, and there are very prescribed ways of handling every single one of these possibilities. And those protocols are maintained by the FDA. Something that I found out today is that right before I connected with Kayla, there was a COVID Q&A between the editor of JAMA, the Journal of the American Medical Association, and the FDA commissioner, Stephen Hahn. 40 minutes of exactly how many different ways they're going to be assessing everything that is happening for prevention or treatment of COVID. And I had never realized, and maybe it's that they're starting it more recently, that all raw data goes to the FDA. So they are continually getting raw data as these studies are going along. And many of the studies actually have an oversight committee that don't know placebo group or treatment group, but they have the results that are being collected. And at any time, they may be able to interfere and open the blinded assignments to be able to look to see whether or not it's a study that may have reached some conclusion that is enough that they don't want to continue it, or that there are significant problems that are occurring. And I actually had not realized that. So what kind of problems can happen after phase 3 when the drug is actually, or the vaccine is actually in the population? And I lived through the 1976 swine flu. I was already a faculty member in a medical school, and I honestly don't remember whether I got the swine flu vaccine shot or not. But the data that I wanted you to be aware of is that it got FDA approval with no problem. 45 million people were vaccinated. 450 of those, approximately 450, developed something called Guillain-Barre disease, or Guillain-Barre syndrome. And of those, there were about 30 deaths. This was not seen in any of the clinical trials, regardless of how big they were, and they all tend to be tens of thousands when they're testing. But Guillain-Barre is an immunologic disease which attacks nerves, and it's ascending paralysis, so that these people were developing first weakness and then paralysis of their legs and then moving up their body. Most of them recovered. One of my colleagues and mentors' daughter, who was a dancer, developed it, and she recovered fully. But I just wanted you to understand that none of these are completely known beforehand, and that's the reason why there are so many issues about safety and group size in order to be able to figure out some of these very, very rare side effects. Next slide. So from the clinical trials, these are the things that I think you should be looking at anytime you see a report about vaccine development or drugs. The first, is it a prospective trial? They developed their protocol was discussed, it may have been amended, many people had input into it, and they laid out all of the parameters, all of the testing that they were going to obtain so that they really could understand what was happening. during the time that patients were receiving the drug or after they received the vaccine. Retrospective means that there's a lot of bias that could be part of that. You could have two patients come in, and whoever was doing the decision making on how they were being treated decided that they wanted to give patient A one of the new drugs, and patient B said, no, I'm not really interested, so they got usual care. Now, you don't know why they weren't particularly interested, and you don't really know the differences in these two people. And so you wind up with two groups at the end that are really just different cohorts, some that may have been in rooms together in the hospital, and one got the drug that they were interested in testing, and the other one didn't. So at the end, you may have two groups that are quite different, and you don't know how that affects the results. That certainly is happening with the treatment of COVID-19. And a number of the trials that are coming out are some patients who got a drug of interest. They may also have gotten a couple of other drugs, but they also got the drug of interest, and then the other group didn't. And you have no idea how to compare the results of those studies. So beware. You want them to be prospective, and you want the patients to be randomized or the volunteers to be randomized. And this is done at a central site, and you have really no ability to change that. Patients will drop out as the studies go along, and most really good studies are compared and analyzed on intention to treat. That's a very specific term that means that you intended to treat all of these patients, therefore you keep them in the study during the statistical analysis. You also want it to be controlled, meaning that if it's a completely new drug or if it's a vaccine, you want it to be compared to placebo, to a group that did not receive the drug or the placebo. And you want to make sure that these two groups now that have been randomized are equivalent for as many characteristics as you could possibly imagine. And table 1 in any publication is the place where all of that information is recorded. And the first thing you should do when you're looking at a trial is look at table 1. They will have one group, they will have the placebo group, and then they will have everybody together. And you can look across, you can see the ages, you can see the difference in number of females versus males, you can see weights, you can see heights, and you can see any number of different drugs or conditions that they may have, and they should be roughly equivalent, which is why big trials make it more likely that you're just looking at the effect of your intervention. And lastly, open label versus blinded. And open label is usual, and as you're going to see in a phase 1 vaccine trial, everybody knows what the volunteers are getting, but you really would prefer in the very large trials where you're comparing vaccine versus placebo or drug versus placebo, that it's double-blinded, neither the patient nor the doctor knows. I pulled up a protocol the other day from clinicaltrials.gov for one of the things we're going to talk about today, and it was listed as quad-blinded. And quad-blinded actually just meant that they were going to be absolutely sure that neither the patient, nor the nurse, nor the doctor, nor the group that was overseeing the study, so that they were the ones that could determine if there was really danger, that they could stop it or they could open it. So that's blinding everybody to who's getting what, so that when they are making their determinants about effect, these are as carefully done as possible and by eliminating as much bias as possible. Next slide.
All right, at this time, we're going to take questions about the clinical trials portion that we just went over. So we have a few questions for you, Shirley. So Lauren asked, how do they identify volunteers for the phase 1 trials? Since this phase focuses on the safety of the vaccine, what happens if the result is unfavorable or leads to death?
Was that 2 questions? First question was how you get the volunteers, correct?
Yes.
Okay. Actually, I've seen a lot of calls for volunteers. If you go to some of the known vaccine development websites, you'll see that they're looking for volunteers. There's also a government website for volunteers that will tell you what clinical trials are in progress and whether or not it's open enrollment, meaning that you could contact them, or whether or not they've already enrolled all the subjects. So there are different ways of knowing about it. Sometimes it's the group of physicians that have been identified as a center for a particular study, and they then know in their community a number of different ways that they can try to get as diverse a population as possible. They may go to church groups, they may go to different types of organizations so that they can increase diversity as much as possible. You want the group to be as much like the population of the entire country as possible. And the second question?
Since phase 1 focuses on the safety of the vaccine, what happens if the result is unfavorable or leads to death?
Hopefully never leads to death. Yeah, and I can't even remember I can't remember in any of the instances that I've been involved with, it was even that. But they start off with a very low dose and monitor just a small number of patients before they then introduce a slightly higher dose. And this happens with drugs and cancer chemotherapy. It really happens quite dramatically that they start with the lowest possible dose. And then as the dose is escalated, as soon as they see an effect, an adverse effect, they will then know that that's as far as they can push that drug. If they are then combining that with another drug, they may back down again to do it. But in vaccines, they start with low dose and they increase, and then based on both the profile of how well the patients are tolerating that, as well as all the laboratory tests, they back down again. And they may not even go any higher if they start, for example, with a low dose and they only do a quarter of the patients in that group and something doesn't look right, they'll just back down. So that's the way it's typically handled.
The next question is from Denise. Is there much communication going on internationally among scientists on vaccine development?
My understanding is yes. The effort from the beginning was that everything be shared as much as possible. And people probably remember that the genome for the coronavirus SARS-CoV-2 was actually published very early in January, within weeks of the time that they identified that as a new coronavirus in China. The entire genome structure of that virus was published. So anybody could start working on vaccine development immediately. And a lot of the other information that's going on is being shared between investigators and all of these little preliminary reports that are coming out. And then in the United States, based on the fact that they've aggregated 5, now 6 companies that are working on vaccine development, there's a lot of information transfer among those groups. I don't know how much, but they're encouraged to provide as much as possible.
We have 2 more questions. A follow-up question from Denise is, do steroids help at all in the treatment of COVID-19?
Yes, and we'll cover that in the next portion.
Okay, and then the last question is from Ellen. I've heard of people getting COVID-19 twice. Since the vaccine would be intended to build antibodies, same as exposure, then how would the vaccine help people can get it more than once?
I think that that's still an open question about whether or not they are getting it more than once. There are tests for the virus that are so sensitive that some of the researchers think that those patients actually are not having a second infection, but they just continue to have symptoms. Because what the PCR test for the virus demonstrates perhaps is dead and pieces of virus that's still left in the system. These are all intracellular viruses that remain. and can then be picked up as they're being expelled from tissues within the body. There are patients that have a very long period of recovery. And I think that at the moment, from everything that I have read, the thought is that they probably have not yet seen somebody who had an absolutely clear second episode. That some people, it lasts for a long time, their symptoms last for a long time, but it's not, and their virus test stays positive, but they're still shedding virus and it's being picked up, not active virus, but degraded virus.
We have one more question actually from Lauren as a follow-up to her first question. Do you think paying patients during clinical trials helps with adherence, or could it sabotage the trial by patients trying to create favorable results?
I really can't answer that. I have no experience with that. None of the patients that I was ever involved with were being paid. One of the clinical trials that I did when I was still in Texas was to test medical residents to look at the effect of stress on their platelets. And this was all part of a NASA grant that I had. And they knew kind of that they had to do it. And when it was all over, they didn't know they were getting paid. We gave each of them a very tiny little gift for it. And that's been my only experience. And I don't know what the volunteers are being paid for these vaccine studies. That's not part of the information that's available unless you're part of this study. They have to be followed for a couple of years. There's a lot of trips to the laboratories in order to have blood studies done. There are a lot of physical exams. One of the things that I heard on the podcast that I was listening to right before I came in here was that now with everyone having smartphones, they are taking symptom information every single day from volunteers, so that when you get up in the morning, you open up your phone and you answer a whole bunch of questions. And so it's easier collecting some of that type of data, but I don't know the answer.
All right, so we'll continue on with the vaccine development portion, and then we will also take another break after this to answer any questions. So you can go ahead, Dr. Levine.
Good, thanks. Vaccines are developed by having an antigen which stimulates an antibody response. And there are many types of antigens that have been used over the years. Viruses themselves can be live, but they're attenuated, meaning that they have been processed in so many different ways that although they're live, they have no ability to cause a disease at that point, or they can be inactivated. And there are some samples I had to look up because I really wasn't sure that I had remembered. Influenza, they have both a live attenuated and inactivated, and I get influenza shots now that I'm older, and I have no idea what I'm getting. Measles, mumps, and chickenpox are all live attenuated, and polio is inactivated. So those were the antigens for those. There are protein-based antigens. They can either be a subunit protein or virus-like particle, pieces of the virus. And polio is the example for the hepatitis B is the example for the protein subunit. And now we get to the two that are really techniques that were not used very often in vaccine development. Something called a viral vector, and I'll explain what that is, or nucleic acid. Viral vector, they actually are taking a virus and stitching it into they're usually adenoviruses that have no activity, so that you can't get a cold if you get that vaccine. But they need something that's going to carry the virus itself into your cells so that you can have an immune response. And there are two of the big vaccine development companies that are using viral vector at this particular time. And they are Oxford Astrazeneca, which is a British firm, but they're part of the group that are being subsidized in part by the United States, or Merck, one of the big pharmaceutical companies. And for the Oxford vaccine, they're actually using a chimpanzee adenovirus and stitching the coronavirus to that directly. And it's all because they have to get these into the cells in order to be able to stimulate an immune response. The last category, DNA or RNA, there's one DNA, and it's not one of the United States ones. But the big one is RNA. And we're actually going to talk about an RNA virus vaccine. There are no approved RNA vaccines yet, which makes everybody very nervous because there have been a lot of people that have tried making RNA vaccines, but for one reason or another, none of them have gotten to the point where they got FDA approval, primarily because they didn't finish the last phase. And I'll tell you a little bit about what I know about that. Mainly the fact that by the time they got the vaccine developed, there weren't enough people with the being exposed to the virus that they could tell the difference between who was vaccinated and who wasn't vaccinated. But they're really the ones that everybody's watching very carefully because they don't want these to fail. So why did they use them? Well, for one, if you're using protein-based, those take years to find the right piece of protein from whatever you're trying to immunize against. May have to change many different times. You may have to go through many different iterations in order to get a protein that is right for stimulating the immune system. And the nucleic acid DNA, RNA are really fast. And that's what I wanted to show you by working through one of the vaccine development processes. And I think most of you know that the United States is supporting a lot of these vaccine development trials. And that's the category that they call warp speed. And there's a lot of money going into it. And one of the things that is quite different and is why there's a lot of money in it is that if the vaccine looks pretty good at the end of phase 1 and part of phase 2, they're starting to make the vaccine already before they even get into phase 3 trials. And they have companies that are promising 1 million doses by the beginning of 2021 or 2 million doses by 2021. All of the developers are associated with several different manufacturing arms. And the goal is to try to get for something that's a pandemic as opposed to, excuse me, the yearly flu shots. They're really trying to make sure that they can vaccinate a whole country and then the whole world with the vaccines that are being developed. And they call that development at risk. And they know that if somehow the vaccine doesn't pan out in phase 3 trial, that's it. They discard all of the vaccine and all of the money is gone. So as we're following all of this, just kind of keep in mind that all the money that they are subsidizing things with is in order to be able to set up the manufacturing at a time when they would not have set it up previously. And they're actually, for some of the contracts that they're signing, they're actually buying 100,000 doses of the vaccine by such and such a date, and the company won't get that money until that vaccine is approved. Next slide. I chose to discuss the vaccine that I first heard about, and it's one of the front runners in terms of how far it has moved along through the pipeline. And because it also has a wonderful story. I think that you may recognize some of these people. I'm not sure who this is, but you can see that President Trump is visiting with Tony Fauci. I'm not sure who that is, and Francis Collins, who is the director of the entire National Institutes of Health. And this young woman is one of the stars, I think, of this entire vaccine development program. Her name is Kizzmekia Corbett, Dr. Kizzmekia Corbett. And she's an immunologist, PhD, and she went to the NIH as a college student and worked in various laboratories. She was in college in Baltimore and won a fellowship to be able to go. And she fell in love with research and research specifically at NIH. She then got her PhD at the University of North Carolina and immediately applied for a job at NIH and was very interested in vaccine development and was hired by the Vaccine Research Center, which is also part of Dr. Fauci's institute, National Institute of Allergies and Infectious Diseases. And when she went into the lab, her supervisor said, "What is it that you want to work on? Do you have any particular virus or bacteria or malaria? I mean, any of the things that are currently being studied and developed?" And she said, "I think I want to work on coronaviruses." And this was about 5 years ago. And she actually spent an enormous amount of time studying at a minute level the virus of MERS, the Middle Eastern virus that followed SARS. And that was she was right in the beginning of MERS and spent a lot of time studying the coronavirus. I knew a great amount about the spike protein, which we're going to talk about in depth. And she was one of the people that was really at the forefront of knowledge about coronaviruses when all of this started. And she has many publications and many presentations that came from her MERS period of time. Next slide. Moderna is a relatively new company, and it's a small company. It's not like one of these gigantic companies like Johnson & Johnson or Merck or Pfizer. It's based in Cambridge, Massachusetts, and they started as a company that was interested in developing mRNA, messenger RNA technology, and they were using it for cancer treatment developments, as well as inherited diseases where they might be able to replace a missing protein in someone. And I think they had maybe one compassionate use product that they got from that. And then also viral vaccines. And the last viral vaccine that I saw that they were working on was Zika. And that only went, I think, up through phase 2, because at the time that phase 3 would have started, Zika was no longer anything that they could find patients that they would be able to study for the active group as well as the placebo group. The CEO of Moderna was on vacation in France when he read about the genome structure of coronavirus. This was January 10th. China had started to have suspicious cases, sort of mid-December to December 20th, December 23rd, that they said looked like SARS, but it wasn't the same virus. And by January 10th, they were able to publish in the literature the exact RNA sequences of that virus. He contacted Tony Fauci at NIH, NIAID, to suggest that they collaborate to develop an mRNA vaccine, mainly because they already were working on a number of projects together. So they already had contracts that they had set up, they knew each other, and it just seemed logical that they would collaborate with this laboratory that knew so much about coronaviruses. And Dr. Kizzmekia Corbett, a senior research fellow at NIH, was working on the coronaviruses, and Moderna knew that. And so because they were already collaborating on other vaccines, they developed their vaccine project. Within 3 days after that genome was published by the Chinese investigators on January 10th, Dr. Corbett and Moderna had already figured out exactly the structure that they wanted for their vaccine. They had finalized the structure completely. And from there, it was, I just wanted to look at the dates because this is something so different for vaccine development. On February 7th, they had their first batch of vaccine that was made. March 4th, the FDA approved their phase 1 study. March 16th, the first patient was injected, and this was part of the Seattle group that had come down with COVID-19, and that was one of the first volunteers at that particular time. So very rapid development, and that's one of the reasons that mRNA technology has really risen to the forefront, because it would take years to do either protein subunits or viruses that had to be attenuated and inactivated and make sure that they were, et cetera. So the only worry was there had never been an mRNA vaccine that had been approved. And therefore, they were taking a huge risk starting with this. Next slide. Okay, so I wanted to go through this because I think that it's such an interesting story, but if I lose any of you, let's answer some things and questions. The investigators select an RNA segment of the virus that contains the receptor, which is called the ACE2 receptor on the spike protein. They know that the spike protein is incredibly important for sticking the virus onto the exterior of all of our cells. And because of Dr. Corbett's work on the coronavirus, they were very familiar with the shape of the spike protein and where the receptors were, and that's what they selected. This is an RNA segment, and they needed to be able to build it out so that they could develop the mRNA from that sequence. And in order to get the RNA, which is usually a very transient molecule as it passes through a cell, they had to stabilize it. So they had to figure out a way to stabilize the mRNA so that nothing would happen to it in the process of getting into the cell and stimulating an immune response. In order to get it into the cell, they had to enclose it in a lipid nanoparticle to get it into the cell. So they've got a lot of this mRNA in a little lipid, tiny little lipid particle, and it's that lipid particle that is able to fuse with the membrane of cells and get the modified RNA into the cell. So now that the messenger RNA is in the cell, what happens? Well, the cell immediately transcribes it so that they make whatever the mRNA is coding for. And in that case, it's the spike protein, and specifically the segment of the spike protein that they developed. And cells start to make spike proteins. Now, this is the perfect antigen. They're all being made as exact proteins, and the cell recognizes them as foreign. So the spike protein, because it is foreign, stimulates a cellular immune response, and antibodies are produced. The plasma antibodies then, as they're circulating in our plasma, can bind to the spike proteins of the virus and block replication and infection. So this is the entire way that these mRNA vaccines work. And I think it's just ingenious, and it's sort of fun to think about the fact that we are making our own body make the exact right protein to stimulate our immune response. Little factories that are busy working. Next slide. So let's just go through the preclinical phase 1 study, and this was just published. Until July, there were some release of information from the company itself, but we really didn't know what was happening with most of it. In preclinical testing, there was full protection against viral replication in the lungs of a mouse challenge model. And yesterday, literally yesterday, or maybe it was the day before, they released information on their non-human primates. And in both of those animal models, they had very good immune responses. The phase 1 interim report, phase 1 began on March 16th, was open label, and those volunteers will be followed for a year. And they used 3 groups of healthy volunteers, all between the ages of 18 and 55, 15 volunteers per group, because the 3 groups were getting 3 different doses, 25 micrograms, 100 micrograms, and 250 micrograms on day 1 and 29. In the preliminary report, And this is published in New England Journal, which is a wonderful medical journal. What they published was that antibody production after the first dose was good, was higher after the second dose, and neutralizing antibodies developed after the second injection to levels in patients with that previously had been infected with COVID-19. So this is a good antibody response, and you're always looking for neutralizing antibodies. You want the antibodies to kill that virus and block proliferation. They say no serious adverse events, and there are books published for the vaccine community that delineates every single category of adverse events, whether it's level 1, 2, 3, 4, et cetera. And it just says no serious adverse events were noted, and that they were mild to moderate. But I don't know if any of you have had reactions to vaccines. I get bad reactions to influenza vaccines, and recently with Shingrix got an even bigger response. But fever, chills, headache, myalgia, pain at the injection site, redness at the injection site, swelling of the injection site were some of the adverse effects that they saw. And these were increased after the second dose. Now, just one comment about first dose, second dose. Most of these vaccines, they would hope to get away with just a single dose, because if they're, let's just say they have a contract for 1 million doses by January 1st. Well, 1 million doses means 500,000 patients. So you always have to think about the fact that 2 doses means a lot more manufacturing per se. And one subject had very early on, with the first dose, had hives or urticaria and was withdrawn from the study. Next slide. So now they moved into phase 2 clinical trial, and the only place that I could really get full information was to go to the clinicaltrials.gov website and specifically look for the phase 2 clinical trial report. These are very succinct reports on exactly what they intend to do. And already, as of May 24th, they've started the phase 2 clinical trial, and I think they've already completed enrollment, so now it's a matter of analyzing it. Double-blind, randomized, controlled trial, exactly what we want. 3 age cohorts, or 2 age cohorts, 18 and 54, and then above 55, anywhere above 55. And patients will be randomized between vaccine versus a saline placebo. Saline is just the salt water concentration that we have in our bodies. And that they're going to be testing 500 micrograms and 100 microgram doses on days 1 and 29. The endpoints, they're going to follow up for a year after the second dose of vaccine, multiple safety parameters, as well as efficacy. And the efficacy is going to be titers of immunoglobulin G to the spike protein by a specific assay, just looking at something directed against another protein, titers of neutralizing antibodies, percent of subjects who seroconverted. Did everybody seroconvert? Did only 80% actually develop antibodies, et cetera? And then some measure of T cell production. They want to know that T helper cells are part of the antibody production, because after the immunoglobulin levels start dropping, one hopes that there's still some T cell immunity that protects subjects and patients. Next slide. Phase 3 clinical trial literally just began on July 27th, and there were articles everywhere about the fact that it was starting. It is a randomized controlled trial. This is where I read in the clinicaltrials.gov report that it was a quad-blinded study. Doctor, nurse, investigator, and the study assessor were all listed in the list of who was blinded. 30,000 volunteers, 18 years and older, and they have settled on the 100 microgram dose. And this particular vaccine is known as mRNA 1273, and that will be given to a certain number of the volunteers and the other placebo group will get saline again. And again, it's 2 doses, days 1 and 29. And they're going to assess the efficacy and safety for 2 years. And they have 89 locations across the United States. This particular company is doing all of their trial studies in the United States, not going out of the country. The Oxford Astrazeneca vaccine is very interesting. Different method, but they also are a forerunner. And they're going down to Brazil, they're going to South Africa, Britain, and some sites, I think, in the United States, but they don't really list the ones in the United States. So what the vaccine companies have to do is look for a place where the infectivity is high enough that they can demonstrate a difference between patients that were exposed and infected, placebo patients, hopefully all, and vaccinated volunteers who have not developed or have developed a very, very mild asymptomatic variety of the disease. So that's the biggest problem when you're dealing with a fast-moving infection, is that in order to test a vaccine, they need to be exposed to the virus or bacteria or malarial mosquitoes, et cetera, et cetera. So that's where we are with the Moderna vaccine. And there were some additional groups that were added to phase 1 that are still to be reported, and that included subjects, volunteers that were in the older age range, and they also had added the 50 microgram dose in between, because they had previously used 25, 100, and 250, and they introduced the 50, and so they were studying that in phase 1. So as you can see, these things move around a little bit. It's hard to exactly pin down what's going on. There will be big reports published, peer-reviewed in journals, so that we can look at everything. But until now, we kind of have to piece together the information from the government documents as well as what's coming out of the companies themselves. And a lot of that, you read more about what's happening to their stock price, going up, going down, going up, going down, than you do about what's happening to the exact trial. Well, you know the start dates, and you kind of know when they've reached enrollment for it. Okay. Kayla, were we going to stop for questions here?
Yes.
Okay.
So we have 3 questions, and then we'll move on to the next question since we're kind of nearing end of time, but we're just going to keep going. So Karina asked, how are researchers taking into consideration physical differences and if this vaccine will impact certain populations differently?
Physical differences meaning what?
I'm assuming as demographics, Karina. I'm not sure if you want to put in the chat what exactly you mean by physical differences.
I think that they're literally making sure that they've got healthy volunteers and subjects, and they're trying to get a very diverse population. And so I'm not sure what the physical differences are, but I hope that they will be doing some of the testing in obese patients, since obesity is one of the major problems in terms of severity of the disease itself, and some of those other things. Age is always an issue. And so table 1, as I mentioned, as soon as the reports come out, tell you exactly how many different characteristics they were trying to get completely randomized in the 2 groups.
Then Cody asked, are the hopes for a widely dispersed vaccine by the end of the year overly optimistic?
I can't answer that. I have heard very good epidemiologists come down on both sides of that. I know that Dr. Fauci is very optimistic, and he certainly knows this particular vaccine development quite well in terms of the responses that they're getting and how hopeful they are that they're getting really good antibody responses. But having said that, think about what it's going to be like for them, for the government, or for the companies, whoever is going to be controlling this, to set up a system whereby certain groups of people get vaccinated and are eligible to be vaccinated. Healthcare workers theoretically should be the very first group. and then to figure out how to distribute and where to vaccinate these people. And something that I haven't mentioned is that there are different conditions under which these vaccines need to be maintained. I remember when I got the shingles vaccine that they had to be kept frozen. And so I couldn't get it at my doctor's office. I had to literally go to the pharmacy where they were able to maintain the vaccine frozen. So there are so many different things that have to be fixed. But just a few minutes ago, my husband was saying that even if the vaccines are finished by the first of the year, we need a little bit more time to figure out safety.
For sure. And I know Denise had a previous question about whether steroids help at all in treatment of COVID. Do you want to say that for the end, or do you want to answer that now?
Right now, because I'm going to finish up with just these three last things that I have.
Perfect.
Next slide. Okay, so drug development for COVID-19. As I mentioned, there are only 2 drugs that have been tested that have demonstrated any response. And you should just wipe away hydroxychloroquine from your brain, because really, really good studies have been done, randomized control, et cetera, that have shown no benefit. So a lot of the stuff that's coming out, it's going to take a long time to figure out whether or not any minor little part of using hydroxychloroquine could be salvaged, and unclear. Dexamethasone is the most powerful of the corticosteroids. And one of the reasons that, well, let's go to the next slide. These are the 3 that I'm going to talk about. There's something called the recovery trial, which was set up in Oxford in Britain. And it's a randomized control trial. And so many patients were being admitted at all times that they decided that they were going to just randomize them between different arms of the protocol so that they could study multiple drugs and placebo. And dexamethasone was one of them. Hydroxychloroquine and which is a malaria drug, anti-malaria, lopinavir and ritonavir are HIV drugs, and neither of them showed any efficacy at all. So that's why they're crossed out. Azithromycin, which is an antibiotic, which was originally given along with hydroxychloroquine in one of the first studies, and I guess that's still ongoing. And then convalescent plasma, where they collect plasma from someone who has just recovered and give it to a patient that's critically ill, because they're passively transferring antibodies to them. They selected these based on a group that knew lots about the different drugs, called the New and Emerging Respiratory Virus Trends Advisory Group. And the way that they chose these drugs was reason to believe that the drug might be effective. The safety profile was well understood. It was available in quantities for 100,000 subjects and could be scaled up to more than that. Next slide. So dexamethasone, which is this powerful steroid, was developed in the 1960s, readily available, any pharmacy. Sometimes I read that it's 6 cents a pill. I mean, this would be wonderful if this turned out to be one of the major ways of treating COVID-19. The reason that they started using it in patients was that it's an anti-inflammatory drug, and you know lots of people that are getting it for asthma and allergies and a variety of other things, collagen vascular diseases. And they tried it in patients who needed oxygen or patients that were on ventilator. And the people that were on ventilator, they've had a real suspicion that the reason that their lungs suddenly turn bad is because they're having an out of control immune response. So they've responded to the virus and then they've over responded. And all of these known cytokines, which are a lot of different substances that are released during the immune response, that they were actually harming the lungs, so-called cytokine storm. What they found was that they had decreased mortality, and they looked within 28 days. There were roughly 2,000 patients that were treated. There were 4,000 some in the usual care group, and it was a statistically significant difference in mortality. Those that were on ventilators had a 1/3 reduction mortality. Those that were just on oxygen had a 1/5 reduction mortality. They didn't see any effect at all on patients who were not receiving respiratory support. So this is the data on dexamethasone and the steroid. And I know from listening to a virology podcast that at Columbia, every patient was getting dexamethasone in order to try to prevent this horrible lung disease with all of their ventilator patients. But this is the study that was done and clearly done in a way that you can trust the results. Next slide. Remdesivir is an antiviral drug. It was first developed for Ebola, but remember, Ebola didn't really come over here very much, but it wasn't even effective when they used it in the Ebola outbreaks. EUA means Emergency Use Authorization. And so remdesivir had never been fully approved by the FDA, but they were able to use it as an emergency drug once the FDA approved it for emergency. They did a double-blind randomized controlled trial that was supported by NIH, but it was done around the world. Very limited amounts of remdesivir, so they divided it up. The company divided it up among many different sections of the world, and patients got this IV for up to 10 days versus placebo. This is a randomized trial. And for patients with lower respiratory tract disease, the lung disease, that had been symptomatic anywhere from 6 to 12 days. The endpoint was the time to recovery, either discharge from the hospital or continued hospitalization, not because they were sick, but because of infection control. Sometimes they develop another bacterial infection that needs to be controlled, whatever. And there were over 500 in the drug group with a median recovery time of 11 days, and over 500 in the placebo group, median time of recovery was 15. So it shortened the time to recovery by 5 days. And this was an instance where the group that was watching safety and efficacy were looking at this blinded and decided that they had to break the code because it looked like there was a group that was doing much better. And therefore, they wanted the placebo group to be able to be started on the drug. So remdesivir doesn't have the same level of efficacy that dexamethasone does for lung disease, but it's still the only drug, the only other drug that has been demonstrated to have any efficacy at all. Okay, next and last, convalescent plasma, which is that plasma collected from recovering patients, can also be collected from patients with something called plasmophoresis, where they remove patients' plasma, return their red cells, so you get a much more concentrated batch of antibodies. Or you can actually make antibodies and give them to the patients. And so this is monoclonal antibody therapy. And there are a number of monoclonal antibodies that are being developed. I chose the one that is now being made by Lilly, but it was started at NIH. It was developed from somebody who was post-convalescent from their blood very early in the COVID-19 epidemic. This was a patient that had developed completely, and they studied his blood and were able to identify a very, very strong neutralizing antibody, and were able to then tease out the cells from his blood that were making that antibody, that specific antibody, and then fuse it with a cancer cell that just grows and grows and grows and grows and grows. And therefore, they have a factory for producing that one particular antibody. And all I could find about it is that their phase 1 trial for using this was started May 28th, and the second trial on June 17th. Phase 1 is hospitalized patients, just 40, and phase 2 is 400 patients. So there's a lot of excitement about the use of monoclonal antibodies because they think not only can they treat critically ill people with them, they were first used, plasma was first used in the flu epidemic of 1918, and it really made a difference in some critically ill patients. And it's been used in other types of infections, And if we can get monoclonal antibodies that can be made by cells that are just little factories of cells that make antibodies that are a clone of cells, that we can not only treat patients, but more importantly, can we prevent? If we have patients that can't get a vaccine because of underlying illness or conditions, would they get this periodically in order to keep them disease-free? Or someone that has an exposure, a well-documented exposure to a family member, could it be given to them before they even know whether or not they're going to get the disease? So I'm ending with a little bit of hope because I think the monoclonal antibodies are going to be ready before the vaccines are. And it's just really nice to know that there's a way to treat this without having to rely on vaccination. We're going to have to learn how to live with the virus for still, I'm afraid, quite a bit more time. Last slide. Okay, can we beat the pandemic? That was my question as I was studying all of this. And can we then share champagne? Will one or more of the vaccines be developed successfully? Will vaccine hesitancy be an impediment? Those patients that are anti-vaxxers, or even people who aren't anti-vaxxers who are just afraid that it's too soon to know all of the adverse effects of the newly developed vaccines. Will adverse effects be minimal and tolerable? What interests me is when you look at some of these trials at the treated patients and the placebo patients, there are as many side effects in the placebo group, the same side effects as in the vaccinated group. Swelling of the arm, pain in the arm, fever, chills, sweats, headache, etc. I saw a study yesterday where there were more of those by percentage of the symptoms in the placebo group who had never gotten the vaccine than the vaccine group. So always important to kind of see what the adverse effects are and look at the placebo group. Will one or two doses be required? As I mentioned, that'll make a big difference in how many people can be vaccinated. Will effective drugs be developed to treat COVID-19 while vaccine development and testing continue? And if so, congratulations to all the scientists, volunteers, patients, and everyone who is working so hard to get herd immunity so we can go on with the life that we remember pre last February, March. And thank you for asking me to do this. I enjoyed really delving into the literature, and I remembered my passion for medicine just knows no boundaries.
Thank you so much, Dr. Levine. We just have two questions that aren't necessarily related to the presentation, but public health as a whole, specific to the career in COVID-19. So feel free to provide any advice to these students. So Tabriz and Salma had similar questions. One of them had asked, what are the perceptions of public health as a profession, and how have you seen that in your career as a physician?
I knew primarily people who were MDs who then got their master's in public health. And I think that that was leading them to a specific area of medicine that would really allow them to look at healthcare delivery systems, et cetera. Or now they're very active. A lot of my favorite people on MSNBC are epidemiologists with an MD degree because, and especially infectious disease specialists. So I don't think I ever really knew any epidemiologists who were just pure epidemiologists. Our paths just really didn't cross. But I think that anyone who's lived through this is going to really value having wonderful epidemiologists, especially physician epidemiologists who can talk to us the way Dr. Fauci talks to us. Or one of my favorite PhD epidemiologist who's in infectious disease is Michael Osterholm, who's at the University of Minnesota. And he has a weekly podcast, which is just so sane, so wonderful, just so knowledgeable that the last 2 weeks he's been talking about reopening schools, which I had to listen to. I've got 8 grandchildren. That was a topic of real interest to me as well. So I think it's a great field. And if someone is in internal medicine or pediatrics and then infectious disease, Master of Public Health is another add-on, sometimes completed during fellowships, subspecialty fellowships. So it's a wonderful way to understand the world a little better.
Yes, a very diverse and multifaceted career.
Yeah.
Salma's question, as a follow-up to that, she mentioned that she's a rising sophomore, specifically a pre-med student. She plans to major in biology and either major or minor in public health. But she wants to have a second plan just in case she doesn't get into medical schools. She's curious about what are the jobs out there for a public health major. She's scared to do the major or minor in public health because she doesn't know really any background knowledge about it, even though she's done a little bit of research. She also wanted to know if you know of any specific opportunities out there in terms of internships and experiential opportunities.
I don't. I'm sorry. My field is so far away from public health as having spent my life in hematology primarily, which was benign and malignant cancer diagnosis in hematology. I think that if she thinks that she's going to love public health, she should do it. I always have, for many years, I was the advisor for third year medical students going through their internal medicine rotations. And they would have to come to me if they were going to apply in internal medicine for internship and residency. And my advice to them always was go to your passion. In the nights that you want to go home and read more about that subject should give you some glimmer of what it is that you really want to do. And if she really thinks that she wants public health, I would say go for it, especially as an undergrad, and see if that's what she likes. And it can't hurt. If she were not accepted to medical school, she could get her master's in public health, reapply. I've seen people do that. They weren't accepted the first go round and they started a master's degree program. Someone that I know very well who was a very good student at Harvard and wasn't accepted to medical school. And I said, what do you love? He goes, oh, I love all the biochemistry. I said, so start a program, a master's degree in biochemistry. And then he wound up at Einstein with me years later, and now has a wonderful career back in Texas and was a star. But sometimes it just takes that little bit of extra. And I think she should find out if that really is her passion.
And the last question we have, COVID-19 has disproportionately affected Black, Indigenous, and people of color. Do you think that this is a clear indicator of the health inequities present in America? And if so, what steps can be taken to address it? And what kind of future changes do you see through health policy?
I think it's going to be tough to really tackle. I think it definitely is health inequity. I spent my entire life taking care of an indigent population, regardless of where I was, whether I was at the county hospital at the University of California as a fellow or in Texas, where the medical school ran the public hospital, where most of our patients were indigent. And then when I came to Einstein, the indigent hematology clinics were the ones that I supervised with all of my fellows. And so I know that group of patients really well. And I don't think they necessarily have the access that allows them to make a decision. Oh, I think I'll go to the doctor and check this out. No, they wind up in the emergency room because of financial health inequities and such. So I think we definitely have to do something. There are lots of people I know that have worked on this, made a little bit of progress, but we need lots of progress in order to make sure that everybody has the same access to health care. And I actually have been somebody who supported a government health care plan. I don't like to see it ushered in without there being some gradual steps moving from the Obamacare type setting to a universal health care. But I think that that's what we need. The people that I know that still have their citizenship in their home countries have universal health care there. It's amazing the difference and sometimes it takes a little bit longer to get appointments in the National Health Service, for example, in Britain. But I had a daughter deliver a baby there that she had things that never could have happened in the United States. She had a 6-week premature baby who was reasonably healthy going home quickly, but underweight, and a HHS nurse came every day with a scale in her hand and weighed that baby for the first week home. I have never seen that done before anywhere. But that was part of their way of taking care of infants in the National Health Service. They had a little clinic that was a couple blocks away, and my daughter could walk him over whenever she wanted to be weighed after this poor visiting nurse had to do it every day, carting a scale around with her everywhere she went. So I'm hoping that as we move more toward a national health service, that we will be able to iron out a lot of those inequities. I hope. And I think that obesity is a major problem here in this country, and I don't know how that's going to be fixed. But I think that since that's one of the major comorbidities in this, I think that it's something that we have to address at the same time. It's part of health. Healthy diet is part of health as well.
Well, thank you so much, Dr. Levine. Thank you to everyone for joining us today as well. For the students, you'll receive a follow-up email, including the evaluation. Once you complete it, you'll receive a confirmation email with the recording link, as well as Dr. Levine, if you're okay with giving out your contact information as well.
Absolutely.
That may want to reach out to you. So we hope that you all join us again for another Women in Public Health Speaker Series presentation. Our next one will be in September 2020 with Dr. Tracy Tavis. She'll be highlighting HIV and aging and how COVID-19 has impacted these populations. Thank you again, Dr. Levine, and everyone enjoy your evening and night.